Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. My guest is Jack Gilbert. He's a professor at UC San Diego School of Medicine. He's a director of the Microbiome and Metagenomics Center. So we're going to talk about intermittent fasting and how it affects our microbiome. So welcome, Jack. Thank you.
Pleasure to be here.
If you would, tell me a bit about your background first, and then we'll talk about the current research you're doing.
Microbial ecologists and microbiome scientists, what I do is I study how microbes interact with each other and how they interact with the world around them. Just so happens that there are a lot of microbes in people and in the world around people, and they shape our immune system and how we process our food and even our mental and psychological capacities. So they're a vital component of our health and wellness. And I got into it by studying microbes and then trying to find new and innovative ways of exploring them.
Okay, well, very good. I've interviewed a lot of microbiologists and spoken about the microbiome a lot, but one thing that occurred to me is what happens when someone's doing intermittent fasting, let's say 18 hours or maybe even 2-3 days. What's happening to all the bacteria that normally would depend on the food we eat? What happens?
We honestly don't know. It's a really understudied area. There have been several investigations, all of them quite preliminary, suggest there's either an increase in the number of bacteria or a decrease in the number of bacteria, an increase in their productivity or a decrease in their productivity. There's a lack of control in the clinical studies that have been performed in this space. What we do tend to understand, though, is obviously microbes exist in their own circadian rhythm, their own day-night cycle, their own understanding of the day, and that they metabolically affects what they do inside the body. And so work that we've done back in the past, about 5-10 years, has demonstrated that there are microbial oscillations that occur through time, especially in a 24-hour cycle or a 48-hour cycle, that seem to affect how the body responds to the world around it, right? So depending upon how you feed, say, a mouse or a rat or a human, you can influence the type of microbial activity that are occurring at different points in the day. We found in one study that eating a high saturated fat, high sugar diet tended to significantly increase the abundance of bacteria that produce things like hydrogen sulfide, the rotten egg smell. And that hydrogen sulfide tended to get it the bloodstream and affect circadian clock genes in your liver. And those genes tell your liver what time it is, right? So eating the wrong kind of food at the wrong time of day tended to produce a microbiome that disrupted how your body sensed time. There is a potential link there.
Well, what about looking at a whole host of biomarkers, before, during, and after a fast? Has that experiment been done? And maybe that would indicate, let's say there's a reduction in B vitamins or serotonin, things that are normally produced in the gut. Maybe that would correlate with those particular type bacteria either changing, function or dying, and other bacteria proliferating and taking over. The same thing with has fecal and oral microbiome been looked at and sampled before, during, and after a fast?
It has, again, in limited studies, and we do see differences, as I pointed out earlier, though. So those differences are conflicting, probably because those studies are poorly controlled or lack some fundamental component that allows us to really interrogate what's going on. But it makes sense, right? What you're saying, the questions you're asked are legitimate. It's just that we don't have the data to support them. It makes that if you turned off the tap of nutrients that support the microbial community in your gut, they're going to have to subsist on other things, right? There are certain types of bacteria, for example, that eat the mucus that lines your intestine. There are certain types of bacteria that survive very, very well and nearly entirely upon the bile acids that are released from your body into the gut cavity. And they can use those as a fuel source. And so, things like, as you point out, it would make a lot of sense to do a clinical study, determine the factors that change before and afterwards, reference to a control population, do a crossover study to determine if those were over time in the same individuals, and see if there were features that could be directly linked to changes in the microbiome. Unfortunately, those studies just have not been done with the rigor that would allow us to make any definitive conclusion.
Well, what do you mean they're contradictory? What kind of information has come out of them? And what's contradictory about them?
Some of the studies have shown an increase in the diversity of bacteria associated with fasting in certain populations. Others shown a decrease. One study in a population of people during Ramadan fasting demonstrated that there was a significant reduction in alpha diversity during the Ramadan period and a significant reduction in the fecal content of short-chain fatty acids. So the concentration of short-chain fatty acids, which are generally generated by bacteria when they ferment fiber, be reduced in the gut or in the fecal matter during the Ramadan period. And that post-Ramadan, when people went back to their more daily consumption of food during breakfast, lunch and dinner, for example, alpha diversity, the number of bacterial species in their food went up and the amount of short-chain fatty acids went up, right? So, saying that this, fasting during the day and eating only after sunset was not necessarily producing what we would normally associate with a healthy microbiome, right? It looked more like an unhealthy microbiome.
Why not look at the metagenomic expression, before, during, and after a fast? Maybe the secret's in there. I mean, there seems to be a lot of redundancy and cross ability, I guess, of various bacteria to perform a certain function. So, in the absence of food, I guess a lot of questions. So that would be one. Is there a change in metagenomics? Is there has anyone, I know it sounds terrible, but, put bacteria under conditions where there's no food for them and to see how long it takes them to die, or if they go into a spore, low metabolic state, or, do other bacteria tend to change function and thereby take over and dominate and take over the production of short-chain fatty acids or whatever it may be, in the absence of food? I guess there's tons of questions there,
I know. Those are all legitimate and very good questions. It's just the studies haven't been done. This is the problem, right? You can do a small-scale investigation with a small number of people and come up with some conclusions, but until you do a rigorous investigation with detailed assessment of the functional potential from metagenomics or the transcriptional activity or the metabolic products in the bloodstream or fecal matter or even the urine of those individuals, and you control the study effectively, you're not going to be able to really identify it. This is why we really do rely on evidence associated with other studies, right? So our study where we look at the scale time effect in a population of animals, right? As we can see that the microbial community does change over time. They do change their metabolic potential. They do change the types of metabolites they generate when you feed them different things. I took away from that study from the University of Chicago that we published in 2016. I took away from that study that you really shouldn't eat a lot of sugar at breakfast, which is the I would have told you anyway, right? You should try and consume instead a high fiber, high polyphenol, fruit and healthy diet in the morning, maybe some yogurt, some granola and some berries, but either way, the problem is the studies haven't yet been done that would allow us to really interrogate that question. What we do know is that intermittent fasting it does have metabolic benefits. People tend to have a reduction in oxidative stress. They tend to have an improvement in their metabolic blood panels. So their cholesterol tends to go down. They tend to look healthier in terms of their blood chemistry. They tend to have lower inflammation. All of those aspects could be associated with changes in the bacterial community. Again, the studies haven't been done to link those two factors, but it would make sense that the microbiome would change. Now, the question you should be asking is, does the microbiome changes associated with intermittent fasting, do those changes lead to changes in the body, or are they just resultant from the body responding to a lack of nutrition at different times of the day? And they're really interesting questions, right? But a lot of it comes down to synchronicity. Your body is in a diurnal cycle. It's in synchrony over time, right? And dealing with...
In the absence of food, okay, so if you have low blood sugar, there's gluconeogenesis that goes on in the liver and other tissues. What about looking at how the body processes change during a fast? Forget about the bacteria for right now, but that may give you a clue. Okay, the production of ketones ramps up, let's say, 18 hours plus into a fast. I'm just making this up. And we know metagenomically that these types of bacteria would probably be able to feed off of them better than these other types. So maybe then we would expect a predominance of them. They would, the mixture would change again during a fast because now the inputs are different. There's not zero inputs, there's still something, but it just changed.
And as I said, we do know that intermittent fasting impacts the immunology. People have less inflammation. They have a better metabolic panel in their bloodstream. They, tend to have less oxidative stress. So people have done detailed assessments of the human being during intermittent fasting, but linking those to the microbial activity just has not been done. But as I said, a lot of those facts are associated directly with changes in the microbiome. Or the microbiome has been associated, microbial metabolic activity has been associated with changes in those features in a person. So it stands to reason there is some kind of association. I mean, again, you cannot just start the microbial community and not expect something to happen. It's supposition as to the mechanism that underpins those associations.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Well, of the studies that you've looked at, does the microbiome come back to the previous state, the unfasted state once the fast ends?
Yeah, but it's been very limited in terms of the number of people that have been investigated. And I wouldn't say they are all robust time series studies where, we're looking at a population before a fast, during a fast, and after a fast with enough samples to really account for the temporal variability, because the people's microbiome changes day to day to a certain extent. And understanding that temporal variability between people has proved to generate really complex understanding. So while again, I referenced that Ramadan study that I read a few weeks ago, but it's a limited study, but it really all it did demonstrate was that the microbiome increased in diversity when people started consuming more food. Would suggest that there is an association between the consumption of a normal frequency diet, an increase in alpha diversity, and an increase in short-chain fatty acid production. But that doesn't necessarily support the conclusions that we see from other studies which show metabolic benefit for the human being from intermittent fasting. So disconnect in what we understand intermittent fasting to do to people, what the limited number of studies that have looked at the microbiome in intermittent fasting have actually shown.
Why do you think there haven't been robust studies? Is it difficult to get an IRB to study this stuff? Is it considered cruel for people to fast, or are there other reasons you think maybe that there's just not enough perceived benefit from doing the work, or what's the reason?
I don't think it's necessarily considered cruel. Obviously, there have been clinical trials that have been performed to assess the actual impact of intermittent fasting. I just don't think, people have applied microbiome investigations to some of those trials, but not enough of them not in a rigorous enough way to really be able to interpret the data effectively. So I don't know, like all of these things, there's only a certain number of people going around that have the ability to generate the data in a way that can actually help to inform clinical outcomes. In the end of the day, we all need more money to do more science, and so there's a certain number of things we can do. Intermittent fasting in and of itself sounds like a great idea, and there looks like there's some perceived benefits. There's a lot of work going on in nutrition and try to understand how to provide adequate and effective nutrition for the individual. I'm chairman co-chair of the Nutrition for Precision Health program for the National Institutes of Health, which is a $175 million program recruiting 10,000 people from around America to investigate or identify if we can determine why people respond physiologically differently to different diets and whether the microbiome might be mediating that. That's a huge investment in trying to just understand how eat affects us. Now, bringing that back down to how the food we don't eat affects us might take some time, right? There needs to be a critical mass of understanding behind those kind of research topics to generate the kind of intellectual investigations that are required to get scientists to behind the idea of investigating it.
In the microbiome, are there predominant species that are in larger numbers that vastly exceed others? Is there kind of a head of the pack? Are there, even though there's however many species and it's diverse, there's certain ones that, again, predominate or dominate?
Yes. I mean, there are organisms which tend to predominate at different times of the day with different types of food in different people, different periods in their lives. And it's a big question, right? There are obviously dominant species and taxa of bacteria, for example, that live inside the body and are evolved and adapted to live inside the body. I mean, one of the most abundant genera of bacteria is the Bacteroides, and they're incredibly diverse inside the gut, and there are lots of different species, and they a lot of different niches inside the ecosystem of the gut, right? But again, if we were to bring it back to intermittent fasting, the changes that we see across the different studies are quite different, suggesting that in the limited number of studies, there isn't a set, type of bacteria that tend to respond to a lack of nutrition going in.
The reason I'm asking is even if one particular species or strain, well, I guess not, there's tons of strains within a species too. What if, I don't know, Firmicutes represents 15% of the total microbial load in a group of people. And then they do this experiment, they just look at what happens to the Firmicutes. Maybe because there's so many of them, changes just in that, that type of bacteria might give a signal instead of trying to look at everything. We're just looking at generic things like quote unquote diversity,
Yeah, I mean, there are going to be metabolic redundancy microbiome that may allow for a certain level of interdependency and resiliency. This manifests from that interdependency. So you can see there'll be ecological stability. I mean, there's some insistence from certain people that intermittent fasting might lead to a resetting of the microbiome. I've seen that talked about. What does a resetting mean ecologically? Well, it means kind of, trying to stabilize the microbial community to a new configuration, right? And that could involve certain microorganisms or a certain diversity of microorganisms that might not be the ones that you had before, right? So you might promote the growth of different strains or different species within a genus or different genera within a family that do kind of similar things, but fill those niches in different ways. And that might be better for you. But again, it's a lot of supposition.
How much will a bacteria's metabolism change in the presence of other bacteria versus not?
For the vast majority of organisms, because every microbe is pumping out products, right? And those products either substantial or secondary food sources for other microbes. It's a cross-feeding orgy in the gut.
Yeah, as I see why it's so difficult to tell. There's just so many, cross dependencies and everything. It's very difficult to sort this out.
Yeah, I mean, if you took it down to a single organism and you said, like you intimated before, you said, if I took a bug and I starved it, what would happen to it? Well, yeah, they do die, right? Cells cannot survive without nutrition. However, they undergo certain periods. So, for example, during surgery, you have to starve yourself prior to surgery, right? And then let's say I'm going to give you gastrointestinal surgery. We're going to do a gastrointestinal resection. I'm going to chop part of your intestine that's got, say, a cancerous polyp on it, right? Then they're going to stitch your gut back together, create what we refer to as an anastomosis. Now, in that situation, I've got to put your body during a fast, right? So you won't be eating for approximately 12 hours prior to the surgical practice because we don't want food in there. We don't want you to regurgitate the food and choke during surgical activity. And then also wash your gut out. If you've ever had a colonoscopy, you'll know what that's like, right? I pump your mouth full of PEG, polyethylene glycol liquid, and you poop out liquid for a period of time until you're nice and clean. Then I pump you full of antibiotic, both oral and IV, and then I cut your gut open and I, chop a piece out. It's a lot of stress, not just for the human body, but for the microbes living in the gut. And one thing we have found in that situation is that people who tended to high saturated fat, high sugar diet to surgery. During all of these insults, the starvation, the antibiotics, the cutting open of their ecosystem and exposure to oxygen, the bugs that tend to thrive and survive under that environment tend to be what we refer to as proteobacteria, so Enterobacter species, and they tend to produce enzymes like proteases or collagenases. And the collagenases especially are a concern because generally in the patients that we get who get infections after surgery, we see that the bacteria causing those infections tend to be ones that can excrete collagenases that break down collagen. Collagen is the protein that builds our body up and makes our cells connect to each other, right? So if your bacteria is producing a lot of that, it can break down your tissue. And so we see this phenomenon, and we have hypothesized that in that situation, this bacteria that produces this collagenase changes its lifestyle when the body starts to suck phosphorus out of the gut cavity in response to all the stress. And that actually causes sudden starvation of that bacteria, and it changes its phenotype to create a biofilm, a living goo, on the inside of the gut. And that biofilm then starts to release collagenase to create nutrient sources out of the tissue lining your intestine. So in that situation, we do see examples where bacteria respond to starvation, hypothetically, in a way that is harmful to our body. Now, that doesn't mean that intermittent fasting is going to cause a lot of bacteria to suddenly emerge, but it does stipulate that under certain circumstances, there could be consequences, microbial consequences to these dietary choices. Or did I lose you?
Has anyone used those pills that have a camera in them, that you swallow to look at the condition of your, of the lining of your intestines and your colon during a fast, or before, during, or after? Maybe that would reveal some clues. I don't know.
Yeah, maybe. I've no idea. I didn't even know they had little cameras in them. I knew they had some that were taking gut contents and looking at the chemistry, but I wasn't aware of little cameras in pills. That's pretty cool.
Yeah, it's a very new thing. Okay, interesting. I guess, yeah, like you said, the experimentation is just still needs to be done to know more. Is there you think there might be a benefit in looking at phage activity? Maybe that's a better indicator of the state of the, of what's going on in the gut, or another window into it, instead of just looking at, the bacteria themselves, or their, the transcriptome or the metagenomics, et cetera.
Yeah, looking at phage activity would be incredibly valuable. Metatranscriptomics would probably tell you something. Again, as I said, metabolomics as well could make predictive changes, and you can look at the bloodstream. It's part, you could throw the kitchen sink at a study, identify changes, and I guarantee you'll see them. Understanding whether any of them are causative or have any relationship to the outcomes that we see in people during an intermittent fast, again, as I say, it needs to be studied with a very rigorous clinical investigation.
What would be a dream study, you think? Or what would be a very rigorous study? Like, what would that look like?
Well, said before, we could do a crossover design where we take people between a standard period of their life, do a full biochemical workup, blood chemistry, gut microbiome, etc. Then put them through an intermittent fast, rerun that chemical profiling, microbiome profiling, and then take them off the intermittent fast for a period of time, rerun the profiling, chemical profiling, microbiome profiling, and then go back intermittent fast. And so you do a crossover design where you see how people respond as an individual to that, and you do that with enough people, and you can build up a statistical power that will allow you to identify any particular trace or biomarkers in the microbiome that might be reflective of the very act of intermittent fasting that might be common across people and not so idiosyncratic, if you will.
What about longer fasts, a week, even a month? Is anyone at all looking in that arena to see what's happening with the microbiome of the people?
Can you fast for a month? Is that
feasible?
Yeah. Oh, okay. Sounds horrible. No.
It sounds really difficult, but yeah, people, a week or 10 days. There's plenty of people I've spoken to that have done that. Yeah, they may do it once a quarter or once a year, but that's, in the bacteria world, that's like eons, I would think. So you might get like a very significant change that maybe wouldn't undo itself after that. I have no idea.
Yeah, possibly it would be able to change back after the food came back. Most bacteria don't necessarily die. They just subsist. They go into quiescent states, or they can form spores and wait to get better. So it's very, very hard to completely change your microbiome. All you can really do is change the proportions of the species. So, if you went on a starvation diet for 10 days, God forbid, it's feasible that you would see significant changes. But when you went back on your diet after that, I would assume that the microbial community would reestablish itself to a very similar level that it was before, assuming you were eating the same types of food before and afterwards. Again, supposition.
Okay. Yeah, no, very interesting. I don't know if, I mean, this is probably not at all linked, but has there been much study on what happens when someone takes antibiotics, to the microbiome?
On the microbiome?
Yeah, before, during, and after, yeah.
Yeah, absolutely. Quite a few studies. Not as, again, lots of studies, and we do know quite a bit, but it depends upon the type of antibiotic people, what they were being treated for, or if they've been treated, and depends upon which part of the gut they're looking in. Different people tend to respond differently. There's some really interesting findings. Invariably, there's one study, which I think is going to be published soon, but where they were able to demonstrate that even after 100 days, high-powered antibiotics associated with the treatment for things like Lyme's disease, people's microbiome after the antibiotics were stopped tended to re-establish in a similar way to what was observed before the antibiotics. So it's to wipe everything out completely. That's probably impossible, but it just knocked it back for a prolonged period of time, and then everything tended to go back to normal. But, in another study, Israel, where they took colonoscopies and endoscopies of the gut or in different compartments all the way through, in a small number of people, because that's a very invasive study. They gave the people antibiotics, and in a certain population, they gave them probiotics after the antibiotics, just a natural lactobacillus cocktail. Those lactobacillus in certain people tended to establish a strong community in the duodenum, which is the part of the intestine just after the stomach, but nowhere else. And you couldn't see those lactobacillus in the stool. You could only see them in the endoscopy of the duodenum. So there's a really interesting problem there. People suggest you should take probiotics after you take antibiotics, but that's really suggested that in certain people, those probiotics could establish in a part of the body they're not supposed to be in, right? Most of the probiotics you buy in the store aren't supposed to live in a human, so they really shouldn't have been establishing in that environment, which is interesting.
I mean, they're not supposed to be living in a human. I thought that, so they're trying to give you novel bacteria and a lot of probiotics instead of ones that are correlated supposedly with good health.
95% of all the probiotics you can buy in the store are food-grade organisms that are associated with fermented foods. So they're bacteria you find in yogurts and, in kimchi and in, whatever. So, I mean, they're generally recognized as safe organisms that we've isolated from food. And instead of consuming the fermented food, we now consume them in pills.
Oh, okay. All right. I thought they were just like wacky ones that were picked out that really had nothing to do with what would normally be seen in a person.
Yeah, you wouldn't normally see these in people, though. They're not designed to live in people. We find them in food, but...
The food, though, I mean, unless, when we eat the food, like you eat kimchi, how much of the bacteria stays in you, or does it all get killed and destroyed?
It's killed and destroyed and pooped out.
You don't think any of it survived?
No, they're not. They don't. Most of them are aerobic. They can't survive in an anaerobic environment like your intestine. So they die and they get pooped out. And you see that because when you stop taking the food, you don't see those bacteria present in the poop after you stop taking the food.
Why would fermented foods be considered to be so beneficial then?
I think because they stimulate the immune system. You're consuming foreign organisms, and your body, like NSA, keeps a record of all the organisms it's seen, and it knows these organisms aren't damaging. It knows they won't survive in our body, and so it calms down when it sees them. It doesn't have an overreaction to them. And so I think there's a immune benefit consuming foreign organisms, either in fermented food or in pills.
Really? That's crazy. I thought that the actual bacteria itself would help you.
That's what a lot of people assume, and that's what a lot of companies will tell you, but we have virtually no evidence of that at all. Only in some of the next generation probiotics, which aren't currently on the market, do we see the bacteria actually performing metabolic activity in the intestine in a robust way that we can actually associate with an outcome, clinical outcome or a health outcome. I don't know any credible solid evidence to support the fact that the bacteria that we would buy in the store actively colonizing your intestine and performing metabolic activity or reactions that are in a living state that support your health. Now, that doesn't necessarily mean they're not health beneficial. As I said, they could be stimulating your immune system even if they're dead. And we found certain probiotics, when consumed by animals, even in their dead, can have very beneficial health effects. So, I mean, they don't have to be alive, but they're definitely not colonizing your intestine.
Well, the ones that are dead, like, the study, I guess, in animals, like what was the mechanism of action by which they helped? How did they help? We
don't know. We hypothesize it's immune activity, as I say. Proving that has proven to be very difficult.
That's so crazy. And that's weird. Well, you ruined one of my, one of my hopes.
Oh, I'm sorry. That was your hope? You could be colonized by foreign bacteria from...
No, but good, quote unquote, ones that would be useful, You eat a ton of fermented foods, supposedly it's correlated with living longer and better.
Well, yeah, that doesn't mean that it's not. The bugs have to colonize your intestine in order for you to live longer and better. This is that's the dichotomy, isn't it? Just because the mechanism of action that you assumed isn't real doesn't mean that it's helpful.
Can someone inoculate their gut, let's say, against C. difficile? Could you take it in food or orally, in certain small amounts, and therefore your body would get maybe used to it, so that if you get messed up later and you would get normally C. difficile taking over, that it doesn't?
No, I would strongly recommend you do not try that experiment. I know. Because you might end up with C. difficile infection, and that would be very, very bad.
Right.
We are very, very diverse peoples, right? And it's possible that in certain situations that certain people have become semi-immune to certain diseases, right? Even when the plague passes through a population, you have people who are immune. I'm sure people are naturally immune to SARS-CoV-2 for reasons that we don't fundamentally understand.
And boy, there's so much mystery. Crazy.
Yes, there is. It keeps me employed.
What is your current research about?
I have 43 ongoing research programs across multiple different disciplines.
But how about one that's super interesting to you that would be fun to discuss for maybe 5 minutes?
Very interested in vaginal atrophy in trans men consuming gender-affirming drugs such as testosterone. So one of my graduate students, Marisol Dothard, came to me with this idea that when women go through menopause, their estrogen level drops. It tends to cause vaginal atrophy, which can cause dryness, which can cause irritation, even bleeding during sexual intercourse. And we are very interested in trying to understand if the microbiome either in the vagina or in the gut was associated with estrogen-based activity that might be either reducing the impact of that phenotype or maybe exacerbating it in certain ways. And so this led us on to try and investigate this in trans men who are undergoing testosterone and gender-affirming therapies. And it's fascinating work because it seems like bacteria in the vaginal cavity potentially release proteases when you consume testosterone that will actually actively degrade the membrane, the mucus and covered membrane that lines the vaginal wall. So it can actually lead directly to atrophy. So the very act of taking testosterone can promote the growth of bacteria, which are detrimental to the vaginal health of trans men.
Interesting. Okay. Well, very good, Jack. What's the best place for people to learn more about your work? Where can they go?
You can go to my website, Gilbert Lab at ucsd.edu. And generally has a lot of the different types of research that we're doing and a link to all the work that we have. It's a good place.
Okay. Yeah. Well, very good. Well, thank you for coming on the podcast. I know it's a lot more questions than answers, but yeah, it's just the more, I guess we look at the complexity of our microbial constituents, the crazier and more complicated it is. So it's going to take a while to tease all this info out if we can.
Yeah. That's science for you. There's always more questions than answers.
Well, very good. Thank you so much for coming on the podcast. I appreciate it.
Liz, Richard, take care.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.